This is number 4 of 18. And so that I want to invite you first off to Lindsay Holson will be talking on Wednesday at 3:10 on composting here in this room, so you're welcome to join that. Also want to invite you all to the chemistry department picnic, which is going to be May 23rd over in City Park. If you are interested, you sign up down by the stock room outside of Daniel's office there, so we have an accurate count of how many people, etc., etc. So, what you said?
Need another minute.
Need another minute. Okay. Are there any questions about that? We usually have hot dogs, hamburgers, gluten-free stuff, vegetarian things.
No hot dogs.
No hot dog. Okay.
Brats, burgers.
Okay. We have pride here. We don't have hot dogs. So if you're hot dog lover, it's not happening. It's an excellent point, sir. I had forgotten about that. Okay. Sorry, I didn't mean to offend.
Heard mine talk about the pigs.
I have.
You're talking about making hot dogs.
Oh, yeah. No, hot dogs are made of like every kind of meat plus other stuff.
Right, but like there are pigs in it.
Are there? Sure.
Might be.
There might be. Okay. There we go.
They're probably. I'm glad I questioned part of the pig.
All right. So we are ready for the introduction. My distinct pleasure to introduce Cyra to you. And so it's been nice. Cyra and I have had to suffer through having her in Biochem 1 and 2 and stuff like that. And then we did independent study on how to kill and cure animals with natural products as well. But probably the most important part about this is she is one of the few Hoosiers here at Lawrence like myself. And so it's so hard to find a basketball-loving Hoosier that comes to Lawrence. Like there's like 4 of us total, maybe even that. Even in the last 10 years while I've been here.
Met like 4 Hoosiers and like the other 3 were not basketball fans.
Right, I mean that's terrible. So we sit back and instead of talking science, we sit back and talk basketball. At any rate, so Cyra's going to be off to Purdue University to my great dismay because I don't like the Boilermakers, and so I'm not going to ask her for basketball tickets. But it's still a very good vet school, which is really important. It is, it is. So without further ado, I'll give you.
So as Dave said, my name is Cyra. I had the pleasure of actually getting a fellowship and working at Purdue last summer before I actually got into the vet school this coming fall. So I got to do my research at Purdue University with a veterinarian by the name of Steve Hoosier, ironically from the state of Indiana. Yes, Dave will like that. So the research that I actually got to do was his question, his idea. So the big idea question was how should we detect and store PRRS. So I'm going to go into what PRRS is, and I'll tell you why it's so important, and the pig idea will make sense. So the first thing we kind of have to know is what is a virus, and that's what the V and the PRRS stands for. So a virus is an infectious agent. So what that means is it's something that gets inside your body and causes a whole bunch of issues. And it can range from like some small things like a common cold, which like we don't think as like that serious, but then like as soon as we start getting to like herpes, well nobody likes having herpes and we start to get a little bit questionable. But then like this winter we heard how bad the flu season was and we were actually having deaths from the flu. So now we're starting to get into viruses that are actually causing human death. And then we get to more serious, deadly things, HIV. So once you're infected with that, eventually, while not immediately, mortality usually happens. So the idea is, in humans, viral infections range from being not so serious to a pretty big problem causing death. However, in animals, it's pretty different. Animals, once they're infected with the virus, usually cause major issues. It's not just like a little common cold for an animal. So it causes big issues in mortality as well as morbidity. And this is important to note because we use animals in our agriculture a lot. If anyone eats meat in here, kind of a big deal. I know not everybody does, but big deal if you're not having as many piglets or as many like little baby chickens show up, then you're having issues getting more product and more money out of it. So some simple, like, non-super serious animal viruses that are still pretty important are like this avian influenza and this equine encephalitis virus, which are in horses and then obviously in birds. But then we move into viruses such as foot and mouth, which is in cows, which is considerably serious. So what happens in foot and mouth is it's a 98% mortality rate. If you go in and like infect 100 cows, 2 of them will survive, everybody else is dead, basically. It has never been in the United States, thank goodness for small things, otherwise our lovely green Packers and cheese would not be a thing because we would have no cows left. So how this kind of applies in life is if anybody has ever gone overseas, typically when you're asked to come back into the United States, you're filling out your paperwork, and one of those check boxes that you have to mark off is whether or not you've been to a farm, which is specifically asking about this foot and mouth disease. Because if you bring it in just from one cow being on a farm, you can have it in your clothes or something, and that could devastate our entire meat agriculture system for cows. So this is a super serious example, but now I'm going to talk to you about the virus that I cared about, which is porcine reproductive and respiratory syndrome virus. So porcine is a fancy way of saying pigs, aka this cute little guy up here. And then this is an actual image of, electronic image of the virus itself. So it gets into pigs, and it actually only needs like 10 little viruses to infect a pig. So we'll talk about how serious this can get. So what are some of the symptoms of this actual virus? So I can list all of these up here, which range from like fever to lethargy, sad, dying piglets, and then piglets that can't survive very well. What does this actually mean? So this is the idea of if you're a farmer that has meat pigs, your pig, if it's infected with PRRS virus, is going to take a time and a half to grow as big as it would have had it not been infected. So instead of your pig growing to its full meat size to have lots of bacon out of it, it's taking 6 months, now you're taking a year and a half to get there. And then on top of that, if you're having stillborn piglets, etc., now you're not even having those pigs being born. So that's putting your time back even farther. So because it causes so many problems for you, for agriculture, everyone that moves a pig or decides to sell a pig or sends a pig off to become meat for people to eat has to be tested for PRRS because it's so serious. So any time that a pig's moved or anything, it has to be tested for this virus. And our process or experiment was to see, could we make testing for this virus better and easier not only for the pig, but also for veterinarians involved testing for the virus. So the virus, a little bit more, it's enveloped. So that was the idea that when we saw that first picture, there was that big blue circle around. It's genetic information, so it has an envelope around it. Its DNA is single-stranded RNA, so as soon as it gets in there, its genetic information can be copied using the host cell's information, or its replication machinery, so it doesn't have to bring in its own, so it can be really small. And there's 2 different types. Obviously, type 1 is in Europe and type 2 is in North America, so obviously we were a little bit more concerned with type 2 since we are in North America. But, so how do we test for PRRS virus? So there's 2 different ways. So in general, when we test for viruses, there's 2 different ways. So if anybody's ever had the flu, you go in, they take a nice little swab, and they stick it up your nose, and then they like put it in a little thing of liquid, and it changes color. That's just telling you that you're actually testing for the actual virus itself. So you're not testing for the immune response. The immune response testing is what is done when you, down here. So testing for the immune response is when you are testing for the antibodies, which we'll go into what that is in a second. But basically what it means is you have to draw blood. So if you've ever had the test here at Lawrence where they test you for HIV, they draw your blood, basically testing for the presence of the virus. So what is the immune response in general? So this picture can be a little scary, but it's relatively simple for what I'm trying to get you guys to understand. So the first idea is that we have this pathogen up here is basically our virus. So our virus gets in, it gets into one of our cells, and the cell breaks it down into little bitty pieces and puts it around its membrane. Once it does that, we now have these cells that are called B cells that have these things called antibodies, which this antibody is what recognizes these little particles of the virus itself. So they recognize, and then over time you develop this memory so that as soon as this virus gets in, the antibodies that are circulating in your blood are like, I know exactly what this is this specific virus, and this is how we get rid of it. So it's your immune system identifying what's going wrong or what's in your body, and then it's like, all right, here we go, this is what we can do. So this is kind of shown better here. Now this picture kind of shows how a vaccine works, but it's the same idea even if you didn't get a vaccine, you got introduced to it to begin with. So the first time you're infected, you have a really small antibody response. So your body doesn't remember it because it's never been exposed to it. But then the second time that you're exposed to it, your body knows exactly what it is and is producing lots of antibodies and is like, yo, body, wake up, we gotta do something about this. This is a serious problem. And then we can get rid of it a lot faster and cause a lot less problems in our pig and or ourselves. So how do we test for PRRS virus? Like I said, there's a couple different ways to test for viruses. PRRS virus specifically draws the blood and draws blood. So there's a couple different ways, and this is exactly where my research started to differ. So the known/tested method is taking about 10 milliliters of blood from a pig, and it's kind of hard to tell in this picture, but it's a really big needle. It's about yay long, and it's stuck up into the jugular of the pig, which is one of your big arteries/veins in your neck, so not great. But my method had the idea of only taking about 250 microliters. Now to put that in perspective, that's about a teardrop of liquid. So kind of the idea of like when a diabetic pricks their finger for blood, or when Shelby, does her bloodletting things, you only need small amounts of blood. So that's kind of the idea that I went with, was only needing this little bit of blood. So once we have the blood, there's kind of two different ways to go with it, and that's the idea of separating the plasma from the blood, and that's the idea of having like your blood cells and your solid parts of the blood separated from the liquid. And you can either do this by centrifugation, which is when we have our 10 milliliters of blood, and I'll explain what that is. And then there was my new method, obviously I'll explain what that is, which are called the Noviplex cards. So when we have our 10 milliliters of blood, we send it down into this little machine that spins it around really, really fast. And the process is called centrifugation. And the supernatant, that's this liquid part up here, obviously is the plasma and the liquid of the blood. And then this little pellet that's made at the bottom are your blood cells. So we're able to get all of the solids in one part and all the liquids in the top part, so we can only extract the liquids when we need it. So what are some of the disadvantages of actually using that centrifugation? So the first big thing is that you actually have to have the equipment to spin it down. So wherever you're at, you have to be able to spin it down. Once you have it spin down, you have to be able to store it, which is this point down here. And to be able to store it and keep it separated, you have to keep it on ice. So as soon as you spin it down, you have to put it in, depending on how many samples you have, a big tub of ice to keep it separated. And then obviously at this point, you also need that 10 milliliters of blood. So we have a test tube sitting in ice, has to be sent off through the mail, which causes issues, when mail carriers aren't careful and we break test tubes, because nobody's ever done that in here before. Causes issues. So then there was my method, and this fancy word called microfluidics basically means that I have this card right here that's able to separate different parts from a liquid. So the idea of taking the blood cells, etc., out of the actual blood itself. So the different parts of the card, first of all, we start with this top overlay. So when you have the card in your hand, there's a top piece that you can peel off, which is what this overlay is. And then you can put your blood from the pig right here on top, and then you let it sit there for about 15 minutes. Now while it's sitting there, it's going through this spreading layer right here where basically it just spreads itself out, kind of self-explanatory, and then it goes through this separating membrane. So that separating membrane captures all of those blood cells, and we're left with the plasma down here. And this fancy down in the collection disc. And then this other isolation screen, basically what that is it's this little Velcro part that keeps that collection disc in place so it's not moving around or falling off. And that's just the base of the card. So then once you're left with peeling that overlay off, you're left with the base of the card, your collection disc, which is stuck onto your isolation screen that has your liquid plasma on it. So from there, now we have what is the big difference between having that big vial of blood and the Noviplex cards themselves? So one big difference is the cost is pretty low. Obviously, you just have to buy a test tube, you need a needle, all of those things can be less than $5. You need your 10 milliliters of blood for your sample, you have to be on ice, and we went over that you also have to have equipment. But this changes when we switch over to the Noviplex cards. So obviously the Noviplex cards save the pigs in the sense that you only have to prick them. Instead of them screaming and you needing headphones or earmuffs to cover up them screaming, it's just a little prick on their ears / bum. Yes. And then obviously you need less blood, and then at this point you're also able to send it through the mail. So instead of having to pay for it to be in this big tub of ice, you not only don't have to pay for that, but you also have the ease of now you don't have to do that, and you can just put it in an envelope, have a 70-cent stamp, put the stamp on, and you can send it off. So it makes it easier for veterinarians to send off samples. And then obviously if we're sending it through the mail, it's okay for it to be at room temperature. Obviously we don't need the ice. And then it just saves time in general. So if you're not struggling with the pig to stick this 10-inch needle in their throat, it goes a lot faster. So once we have that blood separated, now it's time to actually test for the virus itself. And to do this, we use what's called an ELISA. An ELISA is an enzyme-linked immunosorbent assay. So to break that down, basically all we care about is the difference between an antigen and an antibody. So the antigen is the virus. We talked about that, it's just a fancy word. And then the antibody that we went over and the immune response basically is just your immune system recognizing, hey, this is the virus, it's here. And it's in our blood. So the steps of an ELISA, first we have a well plate that's coated with stuff that looks like the PRRS virus. So it's actually not the virus itself, but it's like genetically engineered and placed on that plate to look like the PRRS virus. So the next thing we do is we wash the plate with the pig's blood. So we take the pig's blood from the card and or the test tube and put it on the plate, and then the antibodies in the blood would stick to the viruses that are on the plates. So then you wash it again, and another antibody basically that has this part that changes color hooks onto it. So then what that does is it directly relates the amount of color change to the concentration of antibodies, which is how much the pig's immune system is responding. So the idea is that the more that the pig's infected, or if the pig's been infected, the higher number of antibodies in their system, which means the higher amount of change in color. So for this case, it went from a clear to a blue. So more blue means positive and infected pig. So the results we were looking for, we were trying to find out if can we actually take samples from these cards and test for negative results and positive results? And if we can do that, how long can we leave it on the card? Because if we can only leave it on the card for 24 hours, now we're having to ship through the mail in 24 hours, and it doesn't save us any time or any money because we're paying for 24-hour shipping. So this first graph is just comparing the traditional method, aka your test tube of blood, with taking the blood off of the Noviplex card. And as you can see, the negative from the test tube and the negative from the Noviplex card statistically are the same. So when we had blood from a negative pig, tested negative, great things. And the same thing happened for a positive result. When a pig was actually infected with the virus, both the traditional method and the Noviplex method proved that the pig had a positive result. So this tells us that no matter whether the pig is negative or positive, we can take the sample from the Noviplex card and actually test it from there for the actual presence of the virus. Now that we actually know that the card works, we try to look at how long can we leave it on the card, and do like positive and negative samples vary over time, etc. And we actually showed that both negative and positive samples up here were statistically the same over a 14-day period. So all of these are taken on the Noviplex card, and it's just a positive or negative result the same day, which is what 0 days means. So that means I put it on the card and just tested it right there, 7 days and 14 days after. So what we proved is that after 14 days, we're still going to get negative results and positive results when we should get negative and positive results. So if we have a ridiculously slow mail day, or we get snowed in by a snowstorm, we can still get our samples there within 14 days and we're okay, and we can test the pig for this virus. So conclusions, fantastic, it works. So exciting, obviously the card seems to be able to work within statistical significance, and it can be stored for 14 days. We'd like to test for longer, but obviously 14 days seems like a reasonable time for it to get through the mail. So where can we really go from here? There's a couple different ways. So the first thing is we can test for the presence of different viruses. So the idea is just like we could change the different ELISA test for a different virus and we'd still have that sample from the card and it would do the exact same thing. So we've actually tested it for heartworm, which works, which is also exciting. My partner is actually finishing up his data for that. But we'd like to expand that to more viruses to see if we could get this into more uses. However, the big issue right now is how expensive these cards actually are. The card, one card itself is actually $15 per card, which is not exactly competitive with a 60-cent test tube. Since it is easier, but when you're talking about agriculture and animals, farmers are not willing to pay for a $15 card per each pig because their idea is I need to make as much profit off of that pig possible, and 60 cents sounds a lot better than $15. And the other thing that would be really interesting to go into is to see if we could actually get duplicates off of these cards. So that means get multiple kind of tests off these cards. And the reason that this would be is important is because when you send in a big 10 milliliter vial of blood, you have a lot of blood to be able to run a lot of different things. So if it's, for example, a pig coming in to show, you have to get it tested for 10 different things, and you could all do it from that one vial of blood. At this point, we don't really know how many samples we could take from a Noviplex card and still test positive and negative for each respective test. So that's kind of one area that we really want to look into to see if it's feasible to do that, so then maybe we could convince them to start mass producing these cards and get the price down. But the major thing that we learned from this project, that no piggy should have to go wee 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 all the way home.
Questions?
Yes.
So the 14 days of it not degrading, is that more of a testament to the virus's stability? Is that sort of what you're measuring in the ELISA, is whether or not the virus is present, right?
To a certain extent, of course it does measure the virus's stability, but it's also the idea that even if it's sitting at room temperature, that it's not, that virus isn't falling off the card itself. So it's the idea that that card is able to hold the virus on the card itself. Does that make sense?
Yeah.
So like obviously if the virus didn't have that period of time that it could survive those 14 days, there would be no point in doing 14 days anyway, but it's the idea that it can stay connected to that card for 14 days, that it's not letting it go.
And so just separating the plasma from the rest of the blood cells is enough to make it stable for that long? Like with the other stuff, you have to keep that on ice?
Mm-hmm. That once it's separated itself, because once you pull that top part off, those blood cells are taken away, so you're just left with that plasma. The reason you put it on ice is to keep it from mixing back together. It keeps it separated, so that's why it has to be on ice. Great.
So you say cost is a really big factor. What can you tell me about what they're made out of, these cards? That's really interesting.
Most of it, in and of itself, is obviously cardstock. The separation membrane actually isn't, I can't tell you because he hasn't patented it yet. He is a chemist that works with Purdue, so he hasn't patented it, and he's trying to see if it's worth patenting with his stuff. What I can tell you is that it's not too much more complicated than a piece of paper, because you can do this experiment with like a piece of like just like normal little piece of filter paper. You can separate it out. It's just, it doesn't stay as well on that card, so the idea is that he somehow, the way he's developed it, is that virus stays on there longer than it would just a normal piece of paper.
Follow-up clarification. So you can test for the virus and the antibodies from the plasma? So then when you were, the lecture you were telling us about using the antibodies for the ELISA, but the way you answered Graham and Gus was virus stability.
Yeah. So this gets into the method. So the virus in and of itself is in that plasma sample, but I'm testing for the immune response to that. So they're both caught on that card. The ELISA itself tests for the immune response. If I ran a different test that's called an ELAAS, I could run the same sample from that card and test for the virus itself. So it's just in and of, that card holds both the virus and the antibody, which is the immune response, and then depending on whether you run an ELISA or an ELAS, you can test for either one. The ELLAS just happens to be extremely more expensive, so I wasn't allowed to do that. Why?
So, from my understanding, there's a big push to these microfluidics because of cost reduction. A big part of that seems to be moving the chemist or the biochemist or the vet out of it. And that to me would seem like the most expensive part, even if it costs $15. So you're not, I guess, is this the sort of preliminary step, and then you're going to try pie in the sky goal is it's built in to also tell you and test there, or I guess any idea for that? Because to me it still seems like the most expensive part is built in.
Mm-hmm. To a certain extent, it definitely is, because so the traditional method is you take that vial of blood and then send it off to a lab and they run this ELISA test. So the big difference was the big difference is just that the cost is the convenience of the veterinarian to not have to go out and take blood from the pig. So it could be done by a homeowner, and the homeowner could then send it in instead of having them go to a veterinarian and then sending it off to a tox lab. So that's kind of that big difference in convenience. So you're cutting out that middleman of a vet, having to pay a vet to come out and get it. So you have to kind of weigh that option. So that does make it, like, cuts out the middleman a little bit, if that makes sense. But when a veterinarian comes out, they do take industrial grade earmuffs because the pigs scream so loud. So that's kind of the biggest thing, is that convenience factor there.
Other questions? All right, let's thank our speaker again.